All right. Hi, hello again. We are going to be talking about metabolism, and then we're going to start diving into carbohydrates. So before we get into carbohydrates as one of our biomolecules, we have to talk about the carbon atom and the importance of the carbon atom, because all of these biomolecules contain carbon. And carbon is really interesting because of its atomic structure. So we are going to dive a little into chemistry. So hopefully this should be a reminder for us, or we should remember this from last year's chemistry. But if you look at the carbon atom, you can see that there are 4 valence electrons. And because there are 4 valence electrons, carbon is an extremely versatile element. And you can see here with the Lewis dot structure, same thing. We have 4 valence electrons, which means that carbon can form up to 4 different covalent bonds. That's what I've written right here. So and remember, covalent bonds are when electrons are shared between atoms, in this case carbon, and maybe oxygen, hydrogen. There's a lot of different atoms that carbon can share molecules with. And the thing about the covalent bonds is they're also the strongest type of bond. So because of these outer electrons, carbon is extremely versatile. And what that means is it can bond with a lot of different elements like oxygen, hydrogen, nitrogen, potassium, and other carbons. Because it can form up to 4 bonds, it can create these rings, which we'll talk about in a second, or it can create long chains of carbon, which you'll see in lipids. And it can actually form single covalent bonds or double covalent bonds, which you can see in these 2 examples, methane and then ethylene is right here. These are just 2 examples. So that's kind of the importance of carbon. And then I want to kind of quickly look at some important molecular groups that we'll see in some of our biomolecules. So first one, we have the hydroxyl group, which is basically an OH group. We'll see that a lot when we talk about carbohydrates in just a second. We have these amine or amine groups, which we'll see in proteins and amino acids. That's where amino acids come from, because they have an amine group. Sometimes you may see these carboxyl groups, when carbon is double bonded to one oxygen and then single bonded to another oxygen. So it's a like chew, something like that. And then we have methyl groups, which is when a carbon is bonded to 3 hydrogens, and then, it may be bonded over here. It might be bonded to like another carbon or something or an oxygen or something like that. But, so I might, talk about these certain groups, and we might mention them in class. So just so you are aware of those names and what they mean. So now that we've talked about the importance of carbon, we can kind of talk about metabolism and then go into carbohydrates. So metabolism is not the definition that you probably think of when you think of like your metabolism, how fast you burn calories. Like, metabolism in science, and specifically in biology, has a more general definition. So the actual definition of metabolism is right here. All of the enzyme catalyzed reactions in a cell or an organism. Okay, so normally when we think of metabolism, we think of things like breaking down, especially like your body breaking down and digesting things. That's generally generally what I think of when I think of metabolism, but metabolism is actually the breaking down, but also like the synthesizing of molecules in a cell or an organism. So essentially it's all the pathways in which one molecule or molecules transform into other molecules. So like I said, it can be the synthesis or the creation of large molecules, or it can be the breakdown of large molecules. So sometimes in questions you may see pictures kind of like this right here. So this is an example of a metabolic pathway in the human body, specifically the synthesis of urea in liver cells. So you can see that sometimes they're pathways that actually kind of form more like cycles. So like I said, if you see, a lot of times metabolic pathways kind of look like this. And we'll definitely talk about a lot of examples of metabolic pathways throughout this chapter and then in other chapters. So metabolism is all of the reactions in a cell or organism. And then we can technically break down metabolism into two types. We have, hold on one second. Sorry, I gotta stop. All right, back, sorry. So, like I said, metabolism can be broken down into two types. We have anabolism and catabolism. So anabolism is the synthesis of large molecules, okay? And these happen through the actual reactions are called condensation reactions. And so the examples is pretty much like the synthesis or the creation of most molecules, most macromolecules. So protein, DNA, carbohydrates, like complex carbohydrates. And then photosynthesis is also an example of a condensation reaction because we are building essentially food and large complex carbohydrates for plants. So anabolism is the synthesis of large molecules through condensation reactions. On the other side, we have catabolism. So catabolism is the breakdown of large molecules into smaller molecules. And we do this through hydrolysis reactions. So when we digest and break down our food, that's an example. Cell respiration is the breakdown of large storage molecules into energy. And then when like decomposers digest and break down macromolecules into simple subunits. All of those are examples of catabolism. And the way that I remember catabolism, and thus I can remember anabolism too, I always think, so catabolism starts with cats, and I think cats, they can break things because, cats are a-holes, but cats can break things. And then I also remember hydrolysis goes along with catabolism because lysis, the end of the word, lysis means to break. So you can talk about like cells can lysis, cells can break. So that's how I remember. And then obviously anabolism would be the opposite of that. So that's kind of how I remember those words. That's an easy way to think about it. So moving on to carbohydrates. So this is our first biomolecule that we're going to talk about. And so when we talk about carbohydrates, hopefully you remember this term called a monomer. Remember, monomers are like the building blocks of biomolecules or macromolecules. So carbohydrates, the monomers of carbohydrates are called monosaccharides. Mono means one, saccharides means sugar. So these are the building blocks. They're single sugar units. And the way that you can tell what carbohydrates are, like if you look at a molecule, so a couple things, carbohydrates form rings of carbon, but then you also will find that carbohydrates have 2 hydrogens for every oxygen and carbon. So an example, this is glucose right here, C6H12O6. So you can see that we have 6 carbons and 6 hydrogens, and then we have double that, like double the number of carbons and double the number of oxygens to give us the number of hydrogens. So that's one way, and like I said, we'll kind of look at some examples, but generally carbohydrates have rings of carbons. So going down, we have, the way that it works is we have monosaccharides. So here are some examples, it's not all of them, but some examples of monosaccharides. We have glucose, which a lot of you are probably familiar with, fructose, and then we have ribose. And you might have heard of like ribose before, because ribose is a sugar that's used in like RNA. And then when monosaccharides go through, looking back up here, when they go through condensation reactions, monosaccharides can become disaccharides. So di obviously meaning 2 sugars. So some examples of disaccharides, we have maltose, which is 2 glucose molecules that have bonded. And then we have sucrose, which is a glucose and a fructose molecules that have been bound together. And then disaccharides can then form polysaccharides. Polysaccharides be poly meaning many, so like many sugar units. So starch, glycogen, and cellulose are some examples of polysaccharides, and we'll talk about starch, glycogen, and cellulose in a little bit, and I'll show you what those look like. But before we go into that, I just want to make sure that we know how to draw these 2 specific monosaccharides. So this is something that IB wants you guys to know how to do. They want you to be able to draw glucose and ribose, 2 monomers. So ribose is a 5-carbon sugar, meaning that it has 5 carbons in it. 5-carbon sugar. Okay. So we start off with, technically right now we only have 4 carbons, but there's another carbon, I assure you. So I'm going to do this in a different color. But I'm going to count my carbons. So we always start kind of going clockwise. So we've got carbon 1, we've got carbon 2, this is carbon 3, carbon 4, and then I'll show you carbon 5 in just a second. So, and then we also have an oxygen kind of at the top of our ring. So carbon 1 has a hydroxyl group, OH, and then it also has another hydrogen. And then same thing with carbon 2 and carbon 3, they both also have hydroxyl groups. And then I'm going to draw my little hydrogens, okay? And remember, carbons can form up to 4 bonds, 4 single bonds. So I want to make sure that all of them have 4 single bonds. And then this last carbon right here, carbon 4, it's bonded to a hydrogen, but then it's also bonded to, this is CH8, H, OH. Okay, so again, this is our 5th carbon right here. So that's how we get the 5-carbon sugar. Okay, and then glucose. So glucose is a little interesting. It is a 6-carbon sugar. And technically, there are 2, sorry, I'm going to write this down first. There are 2 forms of glucose. So I'm going to kind of highlight this, but this is, actually, I'm going to draw it right here. So I'm going to count my carbons. So here's carbon 1, we have carbon 2, we have carbon 3, we have carbon 4, we have carbon 5, and we have carbon 6. Okay, and you can kind of see that it's pretty similar to a ribose. Like glucose and ribose are pretty similar. Like most of the carbons, for example, let's see, carbon 1, carbon 2, carbon 3, they all have, they're all bonded to a hydrogen, but then also a hydroxyl group. The only difference that we see is, like, for example, in ribose, they're both pointing down, but then with glucose, we have OH, we have the OH on the 2nd carbon pointing down, but then the OH on the 3rd carbon is pointed up. So that's a little different. So alpha glucose has the hydroxyl group on the 1st carbon. It's pointing downwards, as you can see right here. Hold on one second. So this is the main difference. We have the OH group is pointing down with the alpha, and then beta, it's the same molecule, it's just now the OH molecule is pointing upwards. And I know this kind of looks confusing and probably doesn't make too much sense. So one of the things that we will do in class is we will build these molecules so you can actually kind of see what they, like, how they are different. But the main difference between alpha glucose and beta glucose is this location of the hydroxyl group on carbon 1, okay? So you do have to remember, and you may get a question on the IB exam or on a test or a quiz where it'll simply say draw alpha glucose or draw beta glucose, and you will need to, or draw ribose, and you will have to do that, okay? So these are our monosaccharides. Going into polysaccharides, as I said before, creating polysaccharides is an anabolic process, okay? So, and it's a condensation reaction, okay? And so that means it requires energy. If we're building something, then we're requiring energy. And we're also, as you can see in this picture, I'll make it really big, but what happens is when we have this glycosidic bond form between 2 monosaccharides, we actually, water is produced, okay? So energy is required, but then we also produce water. And the bond that forms between the 2 monosaccharides is called a glycosidic bond, okay? And so, like I said, yeah, and I kind of already said this, and I don't want to repeat myself again. Okay, so that was pretty much all you need to know about polysaccharides, like building polysaccharides. The last thing that I want to talk about is basically the forms of polysaccharides that you need to know. And there are 3, and one of them, as you can see, kind of breaks into 2. So cellulose and starch are both in plants, okay? first thing that we have is cellulose, okay? It's found in plants. You probably have heard of cellulose, or that word maybe sounds familiar to you, but the subunit is beta glucose, okay? So remember, if we go back up, beta glucose, this hydroxyl group is up here, okay? So cellulose creates, beta glucose bonded together with these glycosidic bonds is going to create cellulose. So if you look at this diagram right here, this is showing you that's a glucose, that's a glucose, that's a glucose, that's a glucose. And you can see that, so this is this group right here, this big group, the CH2OH, that group, that's what these guys are. So what happens is when you have these groups, these alternating groups, what eventually actually happens is they create these hydrogen bonds, okay? Because eventually the carbons, so if there's like a carbon, if there are glucoses, beta glucoses up here, and there's beta glucoses up here, they can actually create, let's see if I can make it, like, they create these kind of hydrogen bonds, like that. Okay, so there are all these hydrogen bonds between all of the glucoses from like the first line and the glucoses of the second line, if that makes sense. And so because, remember, we talked about hydrogen bonds are not very strong by themselves, but when there's a lot of hydrogen bonds, it can become very strong. So cellulose is very, very strong. And that's why it's used in cell walls. It's part of cell walls. That's what makes cell walls so strong and rigid and prevents them from bursting. Okay, so cellulose is very important for structure because of those hydrogen bonds. Now, starch and glycogen have different functions. Starch and glycogen are used for storage of energy in the form of glucose, okay? And they all use alpha glucose. So here's our first big difference, okay? Beta glucose is super helpful for creating strong polysaccharides, but then when you have these alpha glucoses, it basically can create these very tight kind of spiraled molecules, as you can kind of see in these pictures below, okay? So we have amylose and amylopectin. The main difference being, so the glycosidic bonds can form on the first carbon and then the fourth carbon. Why does my pencil stop? My keep's not stop working. But anyway, so the bonds can form on the first carbon and the fourth carbon, but then amylopectin and glycogen can have carbons that form between the first and the fourth, but also the first and the sixth. So it can form essentially 2 glycosidic bonds potentially. And so that's why you have this kind of branching happening. So the fact that it can form at 2 places, you have a lot of branches, okay? But they're essentially both starch and glycogen, because of their use of alpha glucose, are really great for storage. Starch is storage in plants, and glycogen is stored energy in animals, but also fungi, okay? Cool. I think that is pretty much it, and we're gonna stop for now. Bye-bye.